Hello everyone. So today I'm going to be guiding you through our Phoretix Toolbox microscopy image analysis software. Now, Phoretix Toolbox isn't strictly just for microscopy image analysis. It can be for any kind of image analysis from any kind of bioimaging device. But microscopy is a market that we've seen quite a lot of uptake of this piece of software. So I thought I'd kind of focus on that for now. It's something that a lot of us as life scientists have experience in. And we like to think that we've got a nice kind of smooth tool, a freeform sandbox, if you will, to be able to form pretty much any kind of analysis that you'd want to be wanting to do. Especially if you're like a startup or in the R&D phase and you don't really know what tools you need yet. You need like kind of a bag of tools to work out exactly what you want to measure. Then Phoretix Toolbox is a really good option for that. Now, I'm showing you the normal TotalLab branded version, but we do have the ability to OEM this product. So if you are a microscopy equipment manufacturer and you're looking for a piece of software to go along with your equipment, maybe you don't have an analysis solution at the minute, please get in touch with us because this is definitely something that we can scale and brand and make it look like your OEM product. So I brought in this image here, which is an example image of some cells. Now at the top here we can see how the software has interpreted the image. Now you know your experiment better than anyone else. So if you need to change any of the contrast or any of the image settings to make that a bit brighter or be able to observe things that perhaps are not observable here, you've got full control over that within this window and you can change all the settings as you want. We've got the ability to zoom, to pan around and fit the image within the window as well. On the left here is kind of our grayscale image view. And over here we have our fully rotatable, fully exportable 3D view of our cells. So if you right click here, you can copy this image to your clipboard or you can save it to a file and then have that, put that in a report, in a paper, a presentation, a poster, anything like that. And what you can also do from this screen as well is you can come through and edit your image. Now, Phoretix Toolbox respects the metadata that you've got associated with your image. So this could be scale information, millimeters, if it's coming straight off a microscope, if that information is within the metadata of the image. Now, editing it within the Phoretix Toolbox application itself preserves that metadata so you don't lose that kind of scale information, the wavelength of light that the image was taken at, transmission or exposure data, all of that information that you might want to keep with your experiment. Phoretix editing within Phoretix Toolbox respects that. So we've got rotates, we've got crops, we've got flips, we've got all the kind of things that you would expect from a simple image editing program. But if you edited this image outside of Phoretix Toolbox in something like Paint, Photoshop, Word even, you would lose that metadata or there's no guarantee of preserving that metadata. So when you came to perform measurements, you might have to get them in pixel lengths rather than in millimeters or inches or whatever your preferred unit of measurement may be. So once we're happy, we've got our image how we want to see it. We've got various different heat maps and colorways here. Once we're happy with the way that we're observing our image, we can then come through and analyze our image. And from here as well, we can add any annotations that we want. So pilots, for example. I've just done that by holding control and left clicking and dragging my annotation to where I want it to be. I can delete it, I can change the font, I can change the text color, the background color, and the pointer color as part of all of this. And if I want to go in and edit one, I can just hold control and double click it again and delete it like that. So we have a number of, like I say, freeform tools to start analyzing loads of different images. So this is my select tool. I don't have any shapes, so I don't have anything to select at the moment. I've got a simple rectangular tool. So if I had some kind of array, you can see here length 0 millimeters, and this is because this image has no metadata with it. But if it did, we could start representing the different sizes of objects. So if you were looking at slides and you wanted to see, the size of a particular stained area versus the size of a stained area or another image, control versus treatment, for example, we can see different things like that. We can also add any kind of coloring to it to differentiate it, and we can also add background subtraction to this shape. So we've got a number of background subtraction techniques here: edge average, surface minimum, shape maximum, and shape mean. So what we can do with that is, if we had lots of different background, if we had lots of different shapes as part of our image, for example, control versus treated, we could take the average, intensity of an area, a cell, or a nucleus or something within our control group and compare it to the average of the treated group to see if our nucleus is growing, our islets are getting larger or smaller, things like that. So we can be really quite multi-layered and quite complex with the things that we're doing within the software, although it seems quite simple on the surface of it. We can add elliptical shapes, again, for the exact same reason. We have freeform polygons, so we can kind of draw around this with our mouse. I'm not going to do the whole shape because it'll be quite boring for you, the viewer. Then we hit complete, and that becomes a shape that we can edit the points of. We can move around if we grab it in the middle. So what you can do is put shapes on draw your shape and then compare it along by dragging it along to your other samples. Again, we've got all the different labels. We can have labels on the bottom. We can change the name of it, so we can say islets, for example. Change all the text color and things like that. So label up our image really nicely like that. If I zoom out again, I can add a grid. So if I'm performing measurements within a grid shape structure, again, if I had some kind of array, or if I had plates with different square wells or circular wells even, with a bit of background subtraction, I could measure their intensity and their area and things like that. You can see I've got all the measurements at the bottom here. We do have this trace tool, which is designed to kind of plop it where you want it, and then by scrolling the mouse wheel or dragging this along, we can kind of grow and shrink the different selections. So you can see here, if I wanted to kind of only select the light area of this image, let's say this was the nucleus, if I change the settings there, hit complete, I can get that shape and start manipulating it just as I can with my other shapes. A bit of a grainy image, so it's not been able to do a great job there with the edging. But if you had a really clear image, that would do a much better job, if you had some kind of fluorescence versus a black background, that kind of thing. We've got the ability to just draw a line across an image. And what we can do with this is we can observe the intensity profile along this line. So you can use this for 1D gels, you can use it for different staining patterns like thin layer chromatography, various different things like that. We can have our polyline. So again, this works like the poly tool, where you can draw any kind of shaped line that you want, complete your shape, and again, you can observe a profile of intensity at the bottom right hand corner here. We can export all of this to CSV, and I should mention that this software works with pretty much any industry standard format. So we're talking TIFF, which is our preferred format for quality reasons. We can do bitmaps, we can do JPEGs, we can do PNGs. I think we can do Fuji file films as well from Fuji equipment. So we really are universal, and we can provide a 21 CFR and GMP compliant version of this software. Again, if you're a microscope manufacturer and you're looking to get into that space and you want to do an OEM deal or something like that, we can definitely brand this up to go with your equipment. We can turn on or off the visualizations of our table and our profiles. And we can, of course, do a curved line like this and start drawing it round, and then fill in the shape like that, complete the shape, and get a really free-form shape there. We can also, of course, get the profile for that shape as well. And again, all of our background subtraction methods apply to all these different shapes. So what I was talking about before is if I say did a multi-select using different selections, I could change I could click and drag these shapes here and set them all as kind of a background for a shape mean. For example, if I just grab one of these, so I can set that as the shape mean and choose that as the background type, and then all of my this shape would be compared to my shape mean of whichever shape I chose. And we can average all of those as well by adding them, of course. You can see them added up here. So this is a quick run through of the kind of flexibility that we can use, that we have within our Phoretix Toolbox software, and the kind of tools that we can offer. It's designed to be basic, quick, simple. That's why it's ideal for R&D scenarios where you're not quite sure where your image analysis workflow should be and what tools you need. We've kind of just given you a bit of everything. So you can export all of this to CSV, you can copy these numbers to your clipboard, and you can change which measurements you want to see on the bottom left-hand side here. You can also export all of this to a PDF report as well, and export all these as presentation images, including your lines and your annotations and things like that. So I hope this is useful for you, the viewer. I hope this will find some use in this product will find some use in some niche applications. By all means, please feel free to click the link in the comment section in the link in the video description to come to our website and get a free trial copy of the software to see how it works within your lab on your images. And please, if there's any tools you'd like to add to this software, or if there's any questions you've got, just leave them in the comments below.